Hi, I'm Lee, CEO of Amaris Bio. We are developing an immunoassay for point-of-care cancer detection. In 2018, cancer was responsible for 10 million deaths. In fact, 1 in 2 of us here will go on to develop cancer in our lifetime. The worst news: half of those who go on to develop cancer will find out at late stages, at stages where cancer is the most difficult to cure. That's because scans are only good at detecting cancer when the tumor is large and more aggressive. What if I tell you today that it's possible to detect cancer even before the solid tumor forms, just by looking at signals in the blood sample? This blood test looks at the DNA that are released by the cancer cell. They are dying using this huge sequencing machine. The problem is, this process takes 2 weeks and costs thousands of dollars. That's because every single strand of DNA in your blood sample needs to be read 15,000 times just so the signal that could indicate cancer could be looked for. Now, if we were to build a tool from scratch, we wouldn't want to build a huge sequencing machine. We would want to build a probe that looked for this keyword right away. And that probe is what we are building. But first, we need to identify what are the mutations that we would like to target. In order to do that, we are using machine learning to identify the data set from thousands of patients. The probe itself is a DNA sequence. And in order to generate this DNA sequence, we are going to use AI based on certain criteria that we specify for. And once the probe is ready, we are going to integrate it with the StackPad technology that has been designed by my co-founder, Professor Robert Marks. So first, a drop of blood is spotted at the bottom of the stack. So this drop of blood may contain cancer DNA and will certainly contain normal DNA. This drop of blood migrates up, and if the cancer DNA is present, it will interact with the probe to form this structure. And the normal DNA will still be free-floating. And they will all travel up until they reach the blocking stack, where the normal DNA will be blocked and will not be permitted to travel further up. And the structure, if present, will continue to travel further up until it reaches the uppermost level, and an enzymatic reaction will happen, and a colored product will be formed. So the formation of the colored product will indicate the presence of the cancer DNA, and the intensity of the color will indicate the concentration of the cancer DNA. This is significant because our solution is 10 times cheaper and 100 times faster than what's currently out there. In addition, it's also scalable because we can make a probe to target any DNA mutations that we want. The big vision is to replace this huge sequencing machine with something that is as easy to use as a pregnancy test, and one that can be used at the point of care. We are in talks with 4 global pharmas and 5 cancer research institutes. And I'm sure you know that there is a huge and urgent need for early cancer detection. As such, we are planning to enter the Chinese market by Q2 of 2021 and the US market by Q2 of 2022. All this will not be possible without a world-class team dedicated to the mission. My co-founder, Professor Robert Marks, is a professor in biosensing and is a patent holder in the biosensing area. He was also a co-founder of 2 biotech startups. I hold a PhD in pharmacology and have 8 years of research experience. And I have also previously founded a robotic startup to automate cancer research. In the future, your blood samples won't take 2 weeks to be processed. It can be processed at the point of care. And cancer screening can be done more routinely. And using our data, pharmaceutical companies can accelerate their drug development process, helping us to find a cure faster. So, thank you.
I think it sounds like a promising solution. What do you think? Questions? Peter, please go ahead.
Oh, okay. Got a chance this time. Okay, look, I do have someone doing cancer research in my lab, but I'm not an expert. But it's my understanding that depending on the tumour, not every... you may not necessarily see signs in the blood, right? Mm-hmm. So presumably this will only be effective for certain kinds of tumour and certain kinds of mutation. So have you decided to focus on specific ones?
Yeah, so this is the problem with the biology, not the technology. So even the biology itself is quite new, this liquid biopsy area. So right now what people are doing is like, there's actually a very big database called the Cancer Genome Atlas. So right now the best thing that we can do is like look at the data from the previous patients and try to deduce like which mutation correlates with which cancer. And also sometimes even if you're positive for a certain mutation, doesn't necessarily equate that you have cancer. So it could be like you need to have like 3 mutations in order to have cancer. Yeah. So yeah, right now I think the best solution is just to look at the data set.
So you have not yet decided to focus on a specific tumour or a specific mutation, is that right?
Yeah, I think like right now which cancer to focus on, it depends on the market, and the biggest market is lung cancer. So like for established cancer like this, we already know like what are the mutations. So for example, in lung cancer, you have the EGFR mutation or the ALK. So by knowing which one is which, then the doctor would be able to prescribe the treatment. So that is why like we have this very urgent need to know which mutation it is. Yes.
Levi, please.
On the plane over here, I watched a very interesting documentary about Elizabeth Holmes. Yeah. You may have heard of her. They raised $400 million, and I think my takeaway, besides all of the other shenanigans, was that it's very hard to do this. So what kind of walls do you have to go through from here to there?
Wow, a lot. But I didn't start with promising like I want to do everything at one time, because I think with Theranos they promised like they would do the hardware and then like they could detect cancer with just a very, very small amount of blood. So in biology there's this thing called the limit of detection. So you need a minimum volume for a reaction to happen. So right now I wouldn't promise that like I just need like one tiny drop of blood and I would do 80 tests at the same time. So with regards to hurdles, so definitely this has to go through clinical trials and then FDA approvals and so on and so forth. Yeah, and also clinical trials with like a very, very large number of samples. Yep.
Any more questions? That might be a good sign. Thank you very much, Wenjing Li.